Uh, Thank you for having me. So my name is Chimwemwe Mhango, and I'm a PhD student at the University of Liverpool, but I'm based in Malawi. So my work is pretty much how we've used some bioinformatics tools to understand the emergence of new rotavirus strains that emerge through multiple reassortment events. So diarrheal diseases account to about the alarms the sixth most cause of death in under five populations. And majority of these infections will normally happen in low to middle income countries. When you look at the etiology of if you look at the etiology of diarrhea, you appreciate that rotavirus is actually regarded as the leading cause of acute gastritis in under five populations. So the main public health approach against rotaviruses has been vaccination, and the WHO has pre-qualified about four vaccines, which over 130 countries are currently using them. And you can actually see the impact in the decline in the number of rotavirus-associated deaths from 500,000 to about 121,000. But the problem with these vaccines is that they don't offer sterilizing immunity. So you still have children being infected by rotavirus, but they only protect against, it generates that initial immunity, so it protects against the severe cases of the infection. Now, this is a problem in its own because this creates an opportunity where these viruses can actually evolve, and you can also generate like vaccine escape strains from the very same viruses that are actually circulating within the setting. So when you look at the biology for these viruses, they're from the Reaviridae family, and they've got 11 genome segments. So each the genome segments will code for like six structural proteins and 5 to 6 non-structural proteins, depending on the virus that you're actually studying. And to classify them, you can do something similar to what they do with influenza, for instance, where you've got like H1N1. So here we use the outer capsid proteins, the VP4 and the VP7, the spikes on the yellow part, to actually classify these viruses. So you can, using the yellow part, which is the glycoprotein, you can classify them into G genotypes or the spikes into P genotypes. But the advancements in whole genome sequencing actually introduced a new way in which you can actually characterize these viruses, where you assign a genotype to all the 11 genome segments. And this gives the virus a true identity of the strain that you're actually dealing with. Now, using that method, you can either, apart from the other capsid proteins, you can either assign a genotype to the remaining 9 segments as well. And when you do that, if you assign them if all the segments are genotype 1, for instance, that's regarded as a wild-like rotavirus strain, because more of these originated from pulsar species. And when you look at, if you assign them like genotype 2, these are regarded as DS1 like rotavirus strains, and they originated from bovine species. But because of the nature, the segmented nature, these viruses will tend to be assaulted during co-infection. So for instance, a single host will have maybe be infected by a virus that has got a wild-like and a DS1 like strain. And then during replication and assembly of new viruses, you find that you get segments from either of the parent strains being assembled into a new single variant. Now that creates a third situation where you have reassortments. And these are easy to pick up if they're coming from like 2 different genotypes. But what happens, let's say if you've got 2 strains that have got the very same genetic, the genotypes behind the genotypes for the rest of the segments, they've got the very same genotypes, but then they co-infect and reassort segments. You're normally going to have issues to actually figure them out. So what we've done with the data that I'm going to show you is how we've been able to tease out some of the within genotype reassortment events that you can actually notice with these viruses. So Malawi actually has one of the longest rotavirus surveillance in Africa. I think it started somewhere around 1997, and so much work has been done. I think clinical trials as well on some of the vaccines that have been pre-qualified by WHO. And what we are currently doing a lot is trying to understand the vaccine effectiveness of the vaccines that we're using against the different strains that are circulating. And previously in our lab, we were able to actually demonstrate how we've had wide genotype diversity of these viruses that have circulated from 1997 to now. And one of the things that we noticed is for this, these G9s were strains that were detected before vaccines were introduced, but then after vaccine introduction, we couldn't see them until like 2017 when they emerged and they became, I think, the third most predominant strain that we're actually typing. So what I wanted to understand is what are the evolutionary drivers behind the emergence of these viruses? So since this is already based on a hospital, it's based on an active hospital surveillance, we just sampled and generated about 13 genomes for these viruses. And I think I'll explain some of the methods that we've actually used in the data analysis when I'm actually presenting the data itself. And we generated these samples, the whole genomes from samples from 2017 out to 2022. So the first thing that we did was to look into what's really the identity of the strains that we're dealing with. So when we did the whole genome sequencing and did the assemblies for these viruses, we noticed that we had about, mainly we had about 2 different variants. The first one was a G9P6, but they had a genotype 2. And normally, this is the one that I was saying was normally it initially originated from bovine species, but then they adapted into the human host and they're able to cause infection in human host. One sample had a wild-like genetic constellation where everything was a genotype 1, but then one of the samples had a mixed infection, which had a G2, G3, and a G9 genotype with both the genotype 1 and genotype 2 genetic constellation. So from this, you can actually see that what we have here, post-vaccine samples are actually predominantly DS1 genetic, they've got that DS1 genetic backbone. But then what I wanted now to look into is why were we having like more samples with a specific genotype than the other samples? So I focused a lot for this talk. I think I'll focus a lot on the 11 samples that had a DS1 genetic constellation. So the first thing that I wanted to do is to check for the genetic variability within those samples that circulated that were detected between 2018 to 2022. So what we did was to calculate the SNP distances between the samples themselves per segment. And then when you look at this, you would actually appreciate that in some samples, you had the SNPs going as high as 300 and 300 SNPs between the different samples that were actually circulating at in that 4-year period. And this was very unusual because the evolutionary rate for rotaviruses, I think, is somewhere between 10 to the 3 and 10 to the negative 3 to 10 to the negative 4, depending on the genome segment. And when you factor that in, we could not explain this. It seemed as if we had like multiple populations in some of those samples. We had like multiple unique populations in some of the segments that we're dealing with. So from this, we noticed that there were some specific segments that had a potential signal for multiple populations. And this is where we started looking into, could there be an opportunity or could that be a chance where we can be able to tease out if within a single host or within our strains that have got a specific genotype, can we be able to tease out if they've got more of like lineages that are different and yet they've got the very same genotypes? So the next thing that we went on to do before we dived into this was to also look at another phenomenon, because previously we noticed that some of the viruses that we studied were actually imported into Malawi. So I just did like a geographical analysis to actually check the samples that we're dealing with. So the first tree is actually just showing you where our Malawian samples are. The blue ones are in the previous samples. I'm not going to focus much on that. I'm only going to focus on the clade that is on top. So when you look at the second tree, you would actually see that within this cluster, our samples were closely related to samples that have been characterized in Mozambique. And this is not new because most of the samples that we deal with, because Mozambique is our neighbor, we normally tend to have strains that are circulating that are similar within the two countries. But one thing that was really interesting was when we started looking at this in the other segments, like the NSP1 and the rest of the other segments that are not the outer capsid proteins, we noticed that the Malawian samples, even though they were clustering together with Mozambican samples, they're also clustering together with some of the previously circulating Malawian strains that were already published. And this was a very new thing because previously we would only find like Malawian samples of what distinct genotypes, depending on the strains that you're studying. So in this case, when we saw this, we noticed that our inner capsid plot and our non-structural genes actually showed a high similarity to previously circulating Malawian strains. So we've got a very rich data set that we've generated. We've got a very rich data set that we've generated previously on whole genome data regarding these DS1-like strains. And this is I think we've generated data from 2000 around 2021. Sorry, I think you'll notice that before 2012, we had fewer genomes. I think it was because majority of the studies that time were not actually generating any genomic data for whole genome data. But after 2012, we actually started sequencing a lot, and I think we've actually generated a lot of data. And when you look at this, you actually see there's a diverse type of, there's a high diversity regarding the samples that we've actually generated data on. So I was trying to leverage this to actually see, can we check if some of the segments that we're seeing, they're actually emerging from any of the strains that we've characterized here. So we did some time-resolved phylogenes for this. And when we did that, the first thing that we noticed was where we're seeing our samples actually cluster. It seemed as if they're emerging from a previously circulating genotype, which was genotype G2. And this was just for the NSP1. But when you started looking at the other segments as well, you could actually tell that in some segments, these samples were actually clustering with genotype 3 samples. So these we're able to actually extract the ancestor data from some of these segments. And when you notice that, for instance, from an NSP1 and NSP2 and some of the segments, you could actually tell that these had an ancestor that previously circulated maybe 2 years before they actually emerged. So from this, we're actually concluding that probably what we're seeing, these strains might have actually gotten their segments from some of the previously circulating strains within a Malawian setting. So what we've done has got a really different, it's got an implication on some of the work that we're doing. So this vaccine that we use in Malawi is a wild-like strain, and it's specific. It's got a better protective, it induces immunity that is better against other homotypic strains, but not strains that are like genotype 2 backbone. Now, we've previously shown that since the vaccine actually works better against the genotype 1, what we're seeing is majority of them as genotype 2. It might be this virus is actually reassorting through complex interactions between the viruses within the setting, and we're generating other new novel viruses. So one of the things that I'm actually now working on is trying to understand how the vaccine actually works against these G9s, and majority of that is actually doing more of like neutralization assays and stuff. This is my presentation, and I'd like to thank a lot of people that I actually work with from different institutions. Thank you.